Hello everyone, welcome. Thank you so much for being here today and joining us here in Southwest Portland to learn more about ALS research, as well as to come together to connect and share experiences and to support one another in living well with ALS. My name is Lance Christian. I'm the Executive Director of the ALS Association Oregon and Southwest Washington chapter. We serve the entire state of Oregon and 6 counties of Southwest Washington. Our mission is multi-pronged. It's to advocate for people living with ALS and for research and public policy reasons, to fund ALS research, and to provide compassionate care and support for families living with ALS. We do that by providing direct support programs to help families every day, but also in partnership with ALS clinics and centers across the region and all the community partners who make a difference in helping families attain and have the resources to live a full life with ALS. A lot of our support programs really range from connecting people with compassionate supporting help, social workers who can do home visits, to help people navigate and plan for the future and the resources they may need, to support groups where people with ALS can come together to meet and connect and share ideas, resources, and experiences, educational events like this one and other ones connected about living well with ALS, things like professional education programs, caregiver boot camps that we do in partnership with Providence to help family caregivers have skills to support their loved ones living with ALS, medical equipment loans for across the entire region in partnership with some of our sponsors that are here today that I'll mention here in a few minutes, as well as assistive technology for computer access and communication support so that people with ALS can always interact and communicate and interact and engage in the world around them, regardless of what symptoms they may experience, whether they're speech problems or functional problems that interfere with people's ability to use technology. So all this means really that we are multifaceted. We want to meet with every family to find out what their experience is and the things that would be helpful to them. So part of our mission as well is to support and fund research to develop new therapies, to find treatments and cures that are evidence-based and have science behind them to show their effectiveness. How we do that with the ALS Association is we are an organization that serves the entire country. We're the Oregon and Southwest Washington chapter, but we partner with our other chapters and our national organization across the country, and collectively we pool our resources together to fund research. And together, each year, the ALS Association funds around $20 to $25 million of research annually. Since the Ice Bucket Challenge, there's been close to over $200 million invested in ALS research. It's quite remarkable. And the prongs of that program are multifaceted. It is understanding the mechanisms of the disease and what's happening in motor neurons and the central nervous system that are part of the disease happening. It's identifying new genes that may be part of ALS. In the last number of years, there's been several new genes that have been identified, partly through ALS Ice Bucket Challenge dollars. And understanding that etiology of the disease leads to the opportunity for smart and brilliant clinicians to work on interventions that may impact to support the continued function of motor neurons, or hopefully in someday to stop the disease. So part of that research as well is supporting new clinical trials, and we're excited that there's a new program that's supporting early clinical trials funded by the ALS Association, as well as looking at environmental aspects that may be part of the disease. So the portfolio is multifaceted, and our goal is to do targeted research that maybe is a little bit not so easily funded by things like the National Institutes of Health or the National Institutes of Neurological Disorder and Strokes to do that early work. And primarily, we also want to encourage collaboration and outside investment. So for every time there is a great scientific outcome at an early clinical trial or development of a new assay or a new theory for therapeutic engagement to treat ALS, it's finding biotech and pharma partners who want to lean in as well. So our goal as an organization is that we are not a bricks and mortar research lab. We don't want to be. We don't care where the science is being done. What we care about is that good science is being done. And so with that in mind, the ALS Association and our dollars contributing into that at this point in time is funding over 150 different research projects, small and large, around the world, in the United States, in Europe, Asia, South America, Australia. ALS is a global disease that impacts the entire world. And so finding the answers to ALS is a global effort. We're thrilled, though, that we have such a great opportunity to have people here locally doing work, as well as across the United States doing phenomenal work to find new treatments to slow the disease and someday stop ALS in its tracks. So I'm thrilled to be back in person this year. We've been doing some virtual research weeks, and we know that for all the people here in the room, we've got multiple more people at home watching our live stream. So we hope today's presentations are illuminating and stimulating and prompt interesting conversations so that we can understand the progress of finding treatment for a cure for ALS. Some of you may have seen this year, sorry, you all got blasted about my work anniversary this week. So I celebrated 20 years with the ALS Association this year. And I can share that what the world looked like 20 years ago, it is night and day. There was one approved treatment with modest benefit. Now there's multiple treatments that are available that are able to be used in combination. The clinical trial world was minuscule, and even the sites where people could access trials were tiny. There really was not a true, there's some very minimal ALS clinic activity prior to 20 years ago, and it really has just blossomed and grown with the creation and support of multiple ALS centers, including our partnerships with Providence and OHSU, who also have the capacity and the ability to offer clinical trials. So it's an amazing difference in what the world looks like. Most recently, since Ice Bucket Challenge, we saw just a plethora of the hundreds and hundreds of more research articles about ALS showing up in the scientific literature. It's quite remarkable. There's a lot of really smart people who are looking at ALS, and for some it's a puzzle, but for many of the people in the room who we're going to hear from today, it's personal because they're treating people with ALS, they have personal connections, and they want to understand and know about the human side of the experience, which is yours, that you bring into the room today. Your voice is as valuable as the scientists, because your experience is your own and it's unique. So we want to thank all of you for being part of this effort as well. It's not just organizations, it's all of you participating in trials and supporting the efforts. We're proud to say that because of the support of our community, in the last 10 years, just right here from Oregon, we've been able to fund an additional $2.6 million in research studies. Two of the presentations today will be by people who we were able to fund this last year, so it's amazing. But none of this happens without you, and it also particularly does not happen without our community partners. Our community partners are really, truly that. They are not just businesses or pharmaceutical companies, they are partners. We are all in this together to make a difference for ALS. And today's event really would not happen without their support. So I really want to thank our sponsors who are here in the room today, and you can see their logos up on the screen and in the live stream. And I look around the room, and some of the folks in these companies have been with us 20 years, and some are newer, but we all carry part of this mission to help people with ALS. So today, I particularly want to start out by thanking the Providence Brain and Spine Institute, which is also the Providence ALS Center. They're one of our premier partners and financial supporters, but also hands-on work, implementing clinical trials and helping people with ALS with treatment and therapies every day. Thank you to the Providence Brain and Spine Institute. Our mission couldn't happen without you. I also want to thank, yes, thank you. Another high-level sponsor today is Mitsubishi Tanabe Pharma America, the maker of Radicava, which is a relatively newer therapy available for ALS. Excited also, we'll hear from one of our presenters about an oral formulation of it. And they're hands-on company that came into the community and has made a big difference and is also trying to continue to fund more research to find new therapies for ALS. We also want to particularly thank some of our assistive technology provider partners. First, I want to thank New Motion, which is a complex rehab company that supports people accessing power mobility and other technology for independence. They support us, many of our loan closets across the state, and they've been partners in many forms for many years, and we're grateful for their financial support. I also want to thank United Access and the Ability Center. They're mobility access transportation specialists for accessible vans and transportation, not just vans, there's lots of options. But hands-on, they are a community supporter that we see that goes out and meets with families and helps them find the transportation solutions that are right for them. So thank you so much to United Access and Mobility Center. Thank you for being here. We also want to give a big shout out and thank you to Norco. Norco is another phenomenal independence partner who supports people with ALS accessing technology for mobility with power wheelchairs, but also they're an amazing provider of respiratory equipment across our entire community. And so many people rely on Norco for respiratory equipment and for AVAPs and trilogies and all the support, as well as the power mobility support. And they are also our loan closet partner in Central Oregon, so we're extremely grateful for their support, and they've been a phenomenal community partner, as well as presenting at many education events. So thank you all for being here. We also want to thank they're not here today, but I want to give a shout out to Pharma, which is a pharmaceutical industry support network who does advocacy in our state. And in particular, I do want to say people think pharma can have sort of a challenging name to it, but I want to say that right now I'm part of a coalition that pharma is part of that's advocating to make sure that people with ALS can get therapies if they happen to have Medicaid, because there's no guarantee that the state will fund those things. So it takes a lot of advocacy work. So thank you to our partners at pharma. And I also want to give a big thank you to Amylyx, who is over here on the left. And they're our newest partner because we're so thrilled and excited that their new therapy was FDA given the thumbs up by the FDA just this last several months. And we're excited to see as this rolls out and people get access to it as another option for treating the disease and slowing progression. So thank you so much to Amylyx and for everyone else for being here. And thank you to all of our staff who are here. Ashley, who's over to show you assistive technology, and our Susan, and all of our staff who are here providing support. So thank you for all being part of this event and part of our community together. So we have several speakers today, and our run of show, first of all, for those who are in the room, a little housekeeping, get up, move around if you need to. If you need to step out, please do. There's a restroom just out the door, a little bit out and down the hallway to the left. But we will be having all of our presenters speak consecutively, and then we will do a panel for Q&A. And when we do Q&A, we will ask, so as you're listening to these presenters, on your table there are notepads or scrap paper. Write down your questions, and we'll gather all those for our panel. So thank you everybody for agreeing to do that. It helps particularly with our online viewers. So our first presenter today, I'm thrilled, is a longtime colleague, and he is the clinical director of the Providence ALS Center, Dr. Nick Olney. He's a neurologist, and he received his medical degree at the University of California, San Francisco, completing his neurology residence at the University of California, Los Angeles. Following his residency, he returned to UCSF to focus on neurodegenerative disease and research. Prior to joining the Providence ALS Center here in Portland, Dr. Olney has a unique, that's where he was before he came to Portland. Nick has a unique perspective in treating and living, supporting people living with ALS, as his father also had ALS. So thank you, and please join me in welcoming Dr. Nick Olney.
All right, hello, I'm Nick Olney. Thanks for inviting me to talk. So here is a picture actually of the sisters there at Black Butte in Oregon, and this is one of my dad's favorite places to visit, both before he had ALS. And when he had ALS, Lance was actually one of the people who drove us down equipment so that we could actually get there and actually be safe in the cabins there. So thank you again, Lance. And when I saw him, when I first interviewed at Providence, I just thought it would probably be a good fit here. So let's see the slides. And they got okay, so this is my dad, and this is me back when I when I was a caregiver for my dad. And I often talk about with my patients that as I was watching my dad with this devastating disease, this is really what kind of inspired me to get into ALS research and learn as much as I could about this disease. In my training, I kind of kept coming back to ALS, and then ultimately came to Providence to be a part of clinical trials, since I do want to be a part of the winning team that will find effective treatments against ALS. So some of my disclosures, I have participated in the Avinar Visiting Expert Program for my work on pseudobulbar affect in ALS. I'm also a PI for multiple clinical trials. I think I'd also just put a disclaimer here that, all this, my talk today is based on data that's currently publicly available, and as research progresses, we're going to know more. We're probably going to even know more in the next 6 months. I'm also really taking a lot of this to highlight, so I'm trying to put a lot of the links and articles as well as presentations where I got these. So if you want to do a little bit of a deeper dive, you can do that later. But I'll try to cover all the kind of greatest hits, so to speak. So before we go into talking too much about trials, I just want to give you a little clinical trials 101. So I think here, if you think of trials in different phases, phase 1 is really the first time in humans. So there's usually been already preclinical data, a lot of lab work, that's a lot of things you're going to hear about earlier. But the phase 1 is usually that first time in humans, often 20 to 50 people. And I would say most of these are usually not coming to the Northwest. We tend to get a lot more of the phase 2 and the phase 3 trials. So the phase 2 trials traditionally would be more set up to test different dosages and to even expand on the safety. And from there we would then get, is there any kind of clinical signal there? And then if it looks good, both safety and there's some signals, we'd move on to phase 3. And that's the phase 3 trials are where you've heard about the drug and placebo. So placebo is patients do not get drug. But then you have to do that, it's required by the FDA to prove that these drugs work. And then they're getting pretty creative with ways of getting earlier access and even mixing placebo into phase 2, so I'll try to highlight that along the way. I do think when someone's newly diagnosed with ALS, I think it's actually very important to stress the importance of the team clinic. I also think talking about FDA-approved treatments is something that's very important. And actually, most of the trials are currently wanting you to decide on your FDA-approved treatments prior to enrolling. So I figured I'd just take a few minutes and at least briefly review some of the data we have for the FDA-approved treatments. And I think it's a very exciting time that patients have this many choices for FDA-approved treatments. So the first compound, Relia-Tech, this first treatment, FDA-approved treatment, going back to the initial trial in New England Journal of Medicine, 1994. These are survival graphs. Here we see basically the survival graph of the upper curve being the upper line being the patients who took Relia-Tech doing better in survival. Also patients here with bulbar symptoms see a little better effect than the ones with the limbs onset. Then if you look at the actual dosing, they moved on to a bigger trial. This is in about 1,000 people, and they did different dosages. So they had placebo group with 50 milligrams on the top, 100 milligrams there in the middle, and then 200 milligrams daily. So then they actually found that the best benefit to risk ratio, because there is a liver effect that we have to look at, liver toxicity, was probably this 100 milligram graph here in the middle. Oops. And this is basically how we came up with the 50 milligrams twice a day for Relia-Tech, which is the first FDA-approved treatment about 20 years ago. So we want to get also away from survival and also looking at actual function. So then the ALS functional rating scale has been created. This is kind of going historically back in time. So this is a 48-point scale. We look at the different regions involved that are typically involved in ALS: bulbar, upper limbs, lower limbs, and respiratory symptoms. And I think one of the coolest studies to come out of this was actually the Radicava study for Radicava. So this was initially looked at in Japan in a big group, and it was hard to show in that bigger group that what worked, but what they saw was a group that was responding, and they repeated that analysis, that trial specifically for that group, and showed a difference, and this is here from the website, of about 30% of the patients who took Radicava doing better than patients that don't. And there's also a good balance between patients who are already on Relia-Tech. I'd also say some evidence that I think is really cool about Radicava, and when patients ask what it'll do for me, is this other paper put out as a post hoc analysis that Jeremy Scheffner did. And this is really showing, so I'm used to this here is, so the open label part here starts at 6 months. So this is kind of the curve we saw before, but this is the group that got placebo. And then when they started taking Radicava, we also see an effect here. So I think that this is one of the things that shows the Radicava helping. And then, as many people know, the oral Radicava came out, and many of the initial side effects to Radicava were the IV, due to the IV infusions, blood clots, et cetera, things like that. So it's really nice to be able to have an oral formulation. A lot of the questions we've been getting in the last month is how equivalent they are. So this again is just showing that the oral and the IV are equivalent. So I think that I've sat through many hours of lectures looking at this data, and it looks pretty convincing. So now we still don't entirely understand all of how Radicava works. So we do have the REFINE study looking at biomarkers. Unfortunately, it does not include drug because it's already FDA approved, so it gets complicated, but it is an exciting study also looking at both oral or IV Radicava. And then the newest kid on the block here, Relivrio, or as I often call it, Amylyx, AMX0035. Just FDA approved in the last month, so exciting to have a third option. So this is Sabrina Paganini presenting evidence from the Centaur study, which is a phase 2 study that included about 140 patients, and they published both in New England Journal of Medicine and in Muscle and Nerve, some findings here. So they showed that at 6 months, the ALSFRS patients did better taking the Amylyx, and I think even more impressive with the survival being 6.5 months longer. They also showed a good safety profile. Predominantly it was GI side effects, probably about a fifth of people. People would know this though in the first 3 weeks. In my experience of patients prescribing Amylyx, probably in the first week. But I think this was a good study. And here's the survival curves again. So the red enriching for patients with the Amylyx and the blue being the placebo. And here's the difference we see over that 6 months. So before screening for a clinical trial, again, I do think it's important to know the FDA-approved treatments and what you want to do, because this is where we have the most, probably the most solid evidence. People are still looking and combing through this data to show exactly, to prove what's happening. So, but I do want to show you some examples of some other studies that have been active in the last year that I've been a part of, and just to give examples of different phase 2 and phase 3 studies. So here was this one. Some of these studies are already enrolled, some are enrolling. But this is AT-1501. This was a phase 2 study, so everyone who enrolled in this study did get drug. It was a dose escalation study that was nationwide. It was 3 months of infusion of this anti-CD4L antibody. And I think another exciting anecdote was this is actually one of the first compounds to move forward in these different phases from the ALS Therapy Development Institute. And this was founded by Jamie Haywood here, who's sitting next to his brother Stephen. And he basically started this company when his brother was diagnosed with ALS to try to get as many compounds into mice and try to actually then get them into actually clinical trials. And there's actually a movie about that called So Much So Fast, and there's a book called His Brother's Keeper. So another phase 3 study that's currently actively enrolling is Combat or Abudilast. It's a phase 2-3 study. So it has actually been FDA approved in Japan, so the safety is pretty well understood. It is looking for a very specific curve in patients with less than 18 months of symptoms. It is a 12-month study with 50-50 randomization. This is Bjorn Oscarsson, who's really the lead on this study. He gives a very cool webinar. Goes over many different ways that suggest neuroprotection in ALS, Parkinson's, MS. There's also many direct and indirect ways that this could be neuroprotective. And I encourage, if you want to know more, there's a good webinar here on the Medica Nova site. Another big trial that we've been a part of and has been pretty popular in the last few months is Courage ALS. So this is clinical outcomes using reldesemtiv on ALS-FRS and global evaluation in ALS. So that's how they come up with that acronym, Courage ALS. So it is a phase 3 placebo-controlled trial. It's internationally trying to recruit 500 patients. It's using reldesemtiv, 300 milligrams twice a day. The mechanism is to slow the calcium and increase muscle contractility. They want to recruit patients within 24 months of first symptoms with breathing above 65%. And where they got this data or how they arranged this trial is based on the phase 2 Fortitude study, which I'll go over in a couple slides here. So Fortitude is a phase 2 study that had some promising data. They used both placebo, so it's a 12-week study, so it's a short study looking for some signal, and they used placebo, 150 milligrams, 300 milligrams, and 450 milligrams twice a day. And then here they actually are showing a difference in that there's less decline in the breathing capacity and the slow vital capacity. They show that again in a different curve. So again, the blue curve is enriched for patients that got drug. And then they also show some changes here in the ALS-FRS. And what they showed was that the best kind of benefit ratio was that 300 milligrams. So that's why they moved forward with Courage. But again, it was such a short study, only 12 weeks. They need to repeat this for a year to actually show that the med is beneficial. So this is an ongoing study right now. It's actually about half filled already. I think another interesting or very novel, unique clinical trial to be a part of has been the Healy platform trial. Now, it's not just one compound. This is multiple compounds all at once, and it has a very novel approach to clinical trials. So for this, let's see, so this is one I was mentioning earlier. This is kind of one of the unique ways of enriching the phase 2 study with placebo to try to see if there's a signal earlier, as well as to give patients more active treatment at the time of the trial. So a way to think about this is in our traditional trials, we kind of set up a giant stadium, we run the race, and then we take the stadium down, and that probably takes about 2 years, sometimes longer, it could be 3 or 4 years. So for the platforms trial, what they're planning to do here is to set up a giant stadium where we can run multiple races at the same time or multiple trials at the same time. And during this, there's better infrastructure, so it cuts cost, it cuts time, and it also allows us to pool the placebo. So to go into this a little bit more, let's see, I was going to use this. So patients come and screen. It's also looking for a much broader population, so allowing patients up to 3 years of symptoms and breathing at about 50%. If they pass into that, they then will get randomized to a regimen. And then the regimen, they come for a specific screen and then get randomized into active drug or placebo. And here we're able to actually pool the placebo, so it's a 3 out of 4 chance of patients getting active drug in the first 6 months. And then all the studies have the ability to expand into an extended access 6 months, after 6 months to being definite drug. I think one of the pros and cons here is that patients don't have a choice on the specific regimen that they're getting, but I think the advantage is the pooled placebo as well as that pooled placebo as well as the access to the drug at 6 months. So another exciting statistic that the Healy has been calculating is they're gearing this to be able to detect something with a 30% change. And what they've already predicted with complicated statistics is they're predicting that with 10 therapies tested, they think they will find this answer. And they'll be using less participants, less placebo, and about a third of the time of a normal traditional trial. Here they're collecting traditional primary endpoints using the ALS-FRS, as I briefly reviewed. They're looking at secondary endpoints of breathing, handheld dynamometry, survival, and they have specific biomarkers for each regimen. There's also ways that, because this is sponsored by Harvard, that people can actually contribute to the Harvard biorepository, and they're also doing some really unique things in virtual monitoring, like with speech and digital, and that really got kicked into the high gear since this kind of opened in the middle of the pandemic. So to give updates, there's actually been results. So I've been talking about this for a few years. When I first came to Providence, we were just starting to try to get this open, try to build the big stadium. And so now we've actually ran the first 3 races. So I want to kind of go over these, the results. So the first one, zilucoplan, which was a complement C5 inhibitor, unfortunately was shut down early due to a futility analysis. So it just didn't look early on like it was doing something. The verdupastat, which is a myeloperoxidase inhibitor, was a negative trial. I think where things start getting interesting here is the CNM-AU8, which was gold nanocrystals, thought to be free radical scavengers and lots of other mechanisms that could work, but there is a bit of a survival signal there, both in the Healy trial, and there was another trial done in Australia showing survival. It's a smaller study and a little bit different population, but I think it's still a potential exciting early finding. So they aren't able to actually show a graph because the pool is placebo, the placebo is pooled, and so we're actually waiting for the regimen D to close to be able to make the actual graph to show that to you. But the predopidine is fully enrolled, and we're actually about to do a database lock any day now. And then the Trihelos is the one that's actively enrolling right now. So this is the presentation that they gave to us about the results of the CNM-AU8. So they had both placebo, 30 milligrams, and 60 milligrams, and the 30 milligrams is where there was the signal of survival. And I think it's a little easier to see here, again, in the Australian study, just because we have that green line, and again, enriched for patients that got drug, and black that did not, but I think that looks like a pretty cool signal. They have had expanded accesses, but unfortunately, I'm not aware that they've gone west of the Mississippi, but I have thrown my hat in the ring to see if they would engage. And then the regimen D, this is predopidine, again, fully enrolled. I think we'll have answers for this by the end of the year, if not early 2023. This is a way that it reduces cellular stress and also helps clearance of toxic proteins. We've got a few patients in Oregon on this compound, so I'm excited to see these results. To talk about the last trial here, regimen E, Trihelos, it's a weekly IV infusion. So unfortunately this one, if you eat the medication, the stomach enzymes break it down, so it has to be an IV formulation. But it's currently actively enrolling. There's multiple mechanisms it could work with protein stabilization and actually helping prevent abnormal proteins from aggregating. It also helps turn on the autophagy pathway in early cell models, as well as help turning on the lysosome pathway. So this was just, again, one of the talks they have on the Healy platform trial. They showed a certain cell model that when they're exposed to trehalose, that it activates the autophagy and increased clearance of the TDP43, which is thought to be the protein spreading throughout ALS. They also have some pretty cool early data on a mouse model, but again, it's a phase 2, so we don't have a lot of data for humans besides that it's basically safe. The one real major side effect is it can raise glucose, so patients with diabetes cannot enroll in this study. And then for one of the last clinical trial stories that I've been following for a long time is the ALS genetic therapy. So I just want to give you some updates on that. So this is something that, first I'll talk briefly about the overall genetics of ALS. I think about 10% we think of as familial or where there'll be a family member that has ALS. But we are finding with the gene C9orf72, that's being found in as high as, this paper is citing 17.2% being familiar with the gene, but I think sometimes even higher than that in some papers. But I'm going to stick with this to mostly talk about the SOD1, because that's the gene we've known the longest about for over 20 years. So when we have a mutation in a gene, we typically have either a loss of function or a gain of toxicity. And we have mounting evidence that the SOD1 has a gain of toxicity. So we think that this is a bad protein and causing damage to the cell. So what we're trying to do with the gene therapies is we use something called an antisense oligonucleotide therapy. So here, I'm going to review just how we get proteins out of the cell, is that we start here in the DNA that makes the RNA, and then that would typically be transcribed into a protein. Now, if this is a DNA with a mutation that we think is causing a bad protein, a way to block that is to insert the RNA that will bind the regular RNA up before it gets translated into a protein to decrease the bad protein. So I actually first heard of this technology when I actually shadowed Tim Miller. This is Tim Miller, so there's a lot of people involved in this technology, but this is where I have a bit of a personal connection because I actually shadowed him in his neuromuscular clinic at my time in UCSD when he was actually learning about this ASO technology in mice before it was really in human studies. But there's lots of different scientists involved in these therapies, but Tim is really one of my main contacts, and he always tends to give a lot of the big clinical talks about this. So the big study that actually went into phase 3 is with the SOD1 ASO called tofersen. It's a phase 3 study that recruited slow and rapid progressors, and it's interthecal delivery. So these are about monthly lumbar punctures. So you'd really want to make sure you have this gene before you'd get this therapy, because it's not always easy to give. But in the last year, they've actually published early results here in the New England Journal of Medicine. Very well respected journal. Just to briefly, just to remind you that the groups were well balanced. There's about 100 people, 60 fast progressors, 48 slow progressors, 72 patients got tofersen, 36 on placebo, and they were both well balanced for Radicava and Riliatec. And so I think first what we're looking at here is the graphs of basically target engagement. So this is actually measuring the SOD1 protein in the spinal fluid. So here is the Valor where it's randomized, and then after this is everybody who's past this graph is definitely getting drug. So what they're measuring here is the red is patients who got the early start tofersen, and the drug and the SOD1 is already going down, and it looks like it's pretty stably down after about 12 weeks. Patients that were getting the late start tofersen here, they didn't go down because they're getting placebo, but then actually when they started getting drug, the SOD1 is going down in a direction we think is good because that's a toxic protein. Another graph supporting of this has good target engagement, again, is with a biomarker we call neurofilament. So neurofilament we think of as a marker in the spinal fluid. It can also be in the blood, but that has been correlated with faster progression or also neuronal death. So we do think that if it's going down, that this is actually a good thing. I do think if in a clinical trial this goes up, that's likely a bad thing. And there's probably still many other mechanisms that could work with neurofilament not being affected, but I think it could guide us if it's going up or down. But here again, we see in the group that's getting the drug early, the neurofilament is going down and staying down. In patients who get it late, the neurofilament starts going down. Now here for the clinical endpoints, unfortunately it did not meet its clinical endpoints looking at the ALSFRS, looking at the 28, but if you recall, the protein didn't really start heading down until 12 weeks. if you start looking at these later data, I do think there's a signal here of something happening. I think you do have to interpret this data with caution, but I think that, and this is ongoing research. But here we see the ALS-FRS. So again, at the 28-week point, unfortunately not significant, but there's still active research going on in the expanded access, or the open label. We see this similar pattern here in slow vital capacity. We see a similar pattern here in the handheld dynamometry, which is again to mimic the muscle strength. We also see, again, survival a bit harder, but it went out quite a bit farther, and we're starting to see some potential signal there. When we look at side effects, again, this is a drug given through lumbar puncture, pushed intrathecally over 2 minutes. Most of the side effects were due to the procedure and headache, and it looked pretty balanced between the groups, because even if you're getting placebo, you're unfortunately still getting the lumbar puncture. So I think in conclusion, I think we were getting good target engagement with the SOD1 and neurofilament going down. At 28 weeks, we unfortunately did not see a significant endpoints, but there is potential effects of earlier versus compared delay initiation of this drug, and further evaluation is needed. So I think that we did also show that in this that the SOD1 is relatively safe, so it is moving on to other studies in asymptomatic SOD1 carriers called the ATLAS study, and I think there is even some talk about the FDA reviewing this data, and again, data for the open label extension is ongoing. So I think, in summary, I think this is an exciting time to be in ALS trials. I certainly, although we're looking at a lot of graphs with downward trends, I'd like someday for these to be flat trends, but we couldn't get this work done without patients and families donating their time. So at this time, I'd like to thank all the patients and families that have contributed to this work. We wouldn't have these answers without that. I'd also think that we're in an exciting time now in clinical trials. I think if you think of the early days of HIV, we would have one compound that would only do something, but when we started getting cocktails, we really started having significant ways of slowing disease progression. And I really do hope that in my lifetime, we can make our ALS centers of excellence ALS treatment centers. And so with that, I want to thank you for your attention and look forward to your questions later.
Thank you so much, Nick. I look forward to the panel because I know I have a lot of questions about how these trials translate into being available as therapies. So I'm excited to introduce an amazing newer member to our community right here in Oregon, Dr. Bahareh Ajami, PhD. She's the Assistant Professor, Department of Molecular Biology and Immunology, and Department of Behavioral and Systems Neuroscience at Oregon Health Sciences University. She's a true international life liver, I just want to say, person who's had broad life experience, did her undergraduate work at Tehran University, got her master's degree at the University of Sydney, and her PhD from the University of British Columbia, and did her postdoctoral work at Stanford University in the Bay Area. Thrilled to meet Dr. Ajami in the recent months. She joined and came to us in Oregon right at the start of the pandemic. So the world shut down, and she was starting her lab. So it's sort of been an honor to kind of connect with her this year, now that the world has opened more up, and hear more about her work. She'll talk a little bit about her scientific investigation, but just she may not mention it, but just to brag on her, many scientists cite other studies within their clinical papers to sort of cite evidence to show the clinical concepts are sound within their work. One of Dr. Ajami's paper from her postdoctoral work is one of the most cited papers in ALS research in many, many years. So please welcome and join Dr. Bahareh Ajami.
Good afternoon. It's really an honor to be here and very tough to follow previous talk. And thank you for a great introduction, very kind introduction, Lance. So as Lance said, I'm a PhD. I do very basic science to try to understand this disease. I'm an immunologist. So COVID was very good for all of us to understand the importance of our immune system. I started my lab in 2019 at OHSU, September 2019. And this is where we are located. I hope I can actually. In RJH, we welcome you to come and visit us at OHSU. This is the lab in our Halloween party, and you all know OHSU and the famous tram. The lab is a combination of immunologists and neuroscientists, and what we're trying to do is basically to understand this disease in component of the immune system. My journey as a scientist, and when did I start to study ALS? As Lance said, I have been in many, many countries. I'm from Iran. That's where I did my bachelor's degree in engineering, actually. Moved to Australia to do my master's degree, and that's where I shift to biology. And was during my PhD, very early time of my PhD, I started working on ALS disease, particularly on immune cells in ALS disease in 2006, when nobody really that much believed that immune cells are important in ALS. And then moved to Stanford to do my postdoc, and then moved to OHSU in 2019 to start my own lab. So what do we study in the lab? We are immunologists, and we are actually located also in the Department of Immunology and interested in immune system. And particularly, we are interested in these cells in immune systems called macrophages. And these cells are the white blood cells that play a very important role in tissue stability and immune defense present in adult tissues. What are their roles? These are the cells that are important for tissue homeostasis. They make sure everybody is calm, and actually very important to guard the tissue. When there are pathogens, they digest them. They clear debris and dead cells. They produce immune messengers called cytokines and chemokines. You heard about that in previous talk, which are very important for their communication and their role. And also, they constantly stimulate other cells in the body, including T cells that previous speakers talk about. One of the most important things to remember is that these cells are very, very first line of defense. These are the cells that evolutionary has been around in every microbe, every living organism. In fact, innate immune cells are the cells that are in plants, they are in flies. Adaptive immune cells, T cell and B cells, are what we have as human, more sophisticated. But these cells are very first line of defense that we have, and our lab is interested to study them in the context of ALS. So we study microglia, which are basically macrophages of the brain. These are the cells that are in a spinal cord and brain. They call microglia, but basically they are tissue-resident macrophages. They possess all the function of macrophages in other tissues that I talked about in previous slide, but they also acquire some specific function that these are the function that is important for them because these are the first soldiers of the brain. So they do other stuff. For example, they're important for synapse formation and pruning and organization of synapses in neurons. They're important for myelin degeneration and formation. Myelin is the cover of axons on neurons, what is causing the communication. They are important for neurodevelopment. And they always interact with other cells in the brain. Astrocyte, neurons, oligodendrocytes. So microglia are our focus as immunologists because these are the only immune cells of the brain. And as I said, I studied them since 2006, and I will take you through the studies we've done and what we are doing right now in the lab. So it's very important to know that these cells react. This is very interesting. Any neuropathologists, when they look at the tissues, they look at how these cells look like because they react immediately when there is an insult. So I hope you can see this movie. This was in 2005, one of the very earlier study that's showing basically if there is a damage, you can see how these cells are start really reacting and going towards the laser capture. So that was the very first time we realized that microglia are actually very surveilling everywhere, and as soon as there is a damage, they go towards it. So why are we interested? Why are we studying microglia? They are the resident immune cells of the brain and a spinal cord. Their main role is to maintain a healthy environment. They are first to react in case of the insult or injury, and they plastic, and this is the key. We can manipulate them. We do need to remember, in a disease like ALS, when neurons, we all know, are post-mitotic, they don't differentiate more. We don't have more neurons. These are immune cells that we could manipulate. And going back to previous speaker that did a great job to setting up this, my presentation is that we believe we can manipulate immune system. This is where we can actually do something. So why microglia are important in ALS? They become reactive following motor neuron degeneration. They are constantly in dialogue with motor neuron and other cells. They become aggregated as the disease happens, and they start having harmful messenger cytokines. They also are these immune cells that possess these tools that could be protective and resolve inflammation. And we can manipulate them to switch them from neurotoxic to actually protective and neuroprotective. So we have these cells that historically, as I put in that one of the very first slides, they are supposed to be responsible for homeostasis. They are resolving inflammation. So we are interested in them in ALS. Why? Why these cells that are actually supposed to do this job, they're not doing it, and how we could actually reverse them. So our previous research, and this is going to touch up a little bit about work I did as a PhD, was to understand basically how these microglia survive during adulthood and neurodegeneration, where they come from. So microglia are cells that are long-lived. Remember just that these innate immune cells in the rest of the body, these monocytes, are not very long-lived. They are 8 to 10 hours. So they constantly replenished by the blood monocytes. However, the stories has always, like the research has shown, these microglia are very long. Sorry, is there a way to go back, hopefully? Yes. So these microglia are showing that they're long-lived. They stay in brain for long, long hours. But when we have neurodegeneration, we have increase in their number. There are new cells coming. And one of the questions during my PhD was that, where are these cells come from? The long hypothesis was that these are the cells that come from blood monocytes, because every macrophages in any other tissue was replenished by blood monocytes. A lot of that point, therapies for ALS was trying to modulate these blood monocytes to send them in, and we were not successful. So when I was doing my PhD, basically we were looking at these green cells, 3 monocytes, to see actually if they come in and replace the microglia in the brain. And our study showed that this is not what's happening. And microglia in the brain are actually inside. They are self-renewing. They're not coming from the blood monocytes, and they actually maintain themselves. So this was the paper Lance was talking about that actually opened up this avenue in the field, that we can't really only focus on monocytes to modulate them. We need to go back and understand microglia itself better in the context of this disease, because they long live. These are the cells that are staying the entire life. And as we know, ALS, similar to a lot of neurodegenerative diseases, is a disease that actually happens when we are aging. Even though if we have that mutation, it doesn't show earlier. So what's going on with these cells? Why these immune cells? This happens to them, and how can we manipulate them? Because clearly they're not coming from monocyte that we could actually change them. So the second question, which I tried to answer during my postdoc, was that what do these cells express? How are they they actually do their function? They are like macrophages. They produce immune messengers called cytokine and chemokine to resolve the inflammation or protect. So the pro-inflammatory cytokine and also anti-inflammatory cytokine. Old hypothesis was that microglia are one population. They either do one job or another. They either have only pro-inflammatory messenger or they only have anti-inflammatory messenger. However, previous studies were looking at these cells similar to these basically Legos as a bulk. They would take all the cells, mush them, and look at them. It's a very exciting time to be a scientist because now we can look at the cells in a single cell level. And around 2012, it was when I started my postdoc and we were able to actually look at these cells single cell, one by one, and actually understand them. And that was very exciting, a study where I started to looking at microglia in ALS, multiple sclerosis, and Huntington disease model in mice, and looking at the cells in a brain and a spinal cord versus blood, and doing this study in a technology called CyTOF that time. It was in 2011, developed by Stanford, and look at the cells one by one and understand, are we really dealing with one cell type that has one messenger, or are we looking at more than that? So the study showed that in ALS model, we actually have 3 different subpopulations of microglia. And there is a population, we call them population B, that is increased in the stage of the disease. And also when we went inside and looked one by one to the messengers of these cells, cytokines of these cells, it was very interesting to see that, for example, you're looking at this population B in end stage, still there are cells that have different messenger. For example, subset of them express IL-10, and I want you to remember this because this is an anti-inflammatory cytokine. But there is a huge increase in TNF-alpha, which is one of the most inflammatory cytokines in this subset at the end. So beginning we came and said there is one population called microglia. Then we discovered there are 3 subpopulations or 3 different microglia population. And again, going inside each, one by one, looking at these cells, we found out there is even more than 3. All of these are based on their function. So now what we have learned from these previous studies that microglia are not coming from the blood, even, sorry, there's a misspelling, neurodegenerative condition like ALS. They reside in the brain and the spinal cord and renew themselves. So if we want to modulate them, we need to find a way to modulate them because we're not going to be able to modulate the one that come from the blood. They are more than one population. And this heterogeneous population has a distinct properties in ALS, as I showed, based on their cytokine production. Our question right now is that, what are the role or dysfunctional role of microglia? Where are the location of toxic or protective microglia? Because I just showed you that we have found a subset that is IL-10 expressing, and the IL-10 is anti-inflammatory. Where is this population? And how we can understand that better, what is the function? And can we use the microglia to protect the motor neuron? So our current study, and I'm going to talk about the 2 projects in the lab, was based of this study, we found one molecule called alpha-5 integrin. Integrins have a history in cancer, also in multiple sclerosis. In this particular integrin is important for migration of the cells, and it seems to be important for migration of microglia inside. So our study showed that when we're looking at tissue from ALS patients, in CD49E, which is alpha-5 integrin, is actually increased in ALS patients. IBA1 is actually a marker for microglia. You can again see this, that it's increased, alpha-5 integrin is increased in people with ALS versus people who don't have this condition. So we did this, also looked at this alpha-5 integrin in tissue from mice, and you can see that alpha-5 integrin in the beginning of the disease, it's not much expressed, but in end stage is expressed in mouse model of ALS SOD1. So it's one of the molecules that is expressed, highly expressed in the end, toward the end of stage in ALS disease and in mouse model. We started treating the SOD, the only mouse model that has the pathological phenotypes, and it's very interesting to see that it's actually increased the survival of these mice with, here, as you can see. So we are very excited about this increase in survival. It's one of the studies in the lab. We hope the publication is going to be out soon, but we also are trying to understand how is this functioning to cause this increase in basically survival. Is it because it doesn't let the bad microglia go for more damage? Another study which we are doing in the lab and we're very excited about is trying to save the neuron by actually getting your lesson from the neurons that are surviving. And this study is based on one of the interesting lessons that the body itself is teaching us. In ALS disease, even until the very, very end, there are certain motor neurons that are spared. In fact, you can see here, ocular motor neuron, Unfus nucleus motor neuron, are resistant to this disease. And we need to remember, this is even true for people who have the mutation. What is the difference between those motor neurons and the motor neurons that are actually degenerating? And we believe the answer is in microglia. Basically, we believe that the cells, microglia cells that are around this ocular motor neuron are protective, that the ones that are around the degenerative are the neurotoxic microglia. And we're trying to understand what is the difference between these two, and can we later on, similar to the previous speaker, actually reprogram and causing this neurotoxic microglia becoming similar to the other one. So what we are doing, we are taking advantage of what the technology is offering right now. Basically studying these cells one by one on a single cell level to understand what is the difference between a microglia around a protected neuron versus a microglia around a degenerated neuron. Our current study is basically, we are trying to profile, and this is one of those studies, as Lance mentioned before. NIH would not fund these things because we are still fishing for those molecules. So we understand the profile and location and the function of the protective and toxic microglia, and then we're trying to manipulate this to stop the degeneration. How are we doing this? We are using two technologies called the spatial transcriptomic. Basically, we are going in the tissues and looking at the genes around those neurons from the microglia. We also are using single-cell transcriptomic, and both of these together is showing us where microglia profile around the degenerated neuron versus protected neuron. This lens to be able as a biologist to go and look, it's amazing. It's an unprecedented, and I feel very privileged to study in this era, this science, because the technology just started in 2020. So it's been very amazing to be able to go one by one, look at the cells, look at the neuron next to them, and understand what's happening. And I just touch base on a very small amount of data we have from this study. So similar to I showed you there was a IL-10 that we were finding during my postdoc that it was very surprising. Even at end stage, we have anti-inflammatory. And the hypothesis started from there. Is this IL-10 expressing microglia around the protected neurons? And recently, with this study, we also have been able to find that in ocular motor neuron, are where the IL-10 expressing the genes down the pathway of IL-10 is actually upregulated, as you can see in ocular motor neuron as onset. And basically, we believe that it's one. The beauty of these data sets are that this is one that is confirming there is a difference around those ocular motor neuron in microglia. They are protective. They have certain messengers that protect them. So basically, IL-10 is an anti-inflammatory immune messenger produced by microglia that reduce inflammation by inhibiting the production of inflammatory immune messengers. And basically, it actually reduces the impact of the inflammation and exacerbation of the motor neuron degeneration. It's an early time of this research, and we are hoping the more we're looking at these two different areas, we're doing both in human and mice, we'll be able to actually understand better how this protection happen and how we can learn a lesson from that and go back and protect the rest of the neurons. So IL-10 right now is our foremost candidate, but we are actually hoping to have more and more. This is a huge data set. We have almost 2,000 genes that we are actually going through and finding. But I want to talk also as a patient. In 2020, I was diagnosed with triple-negative breast cancer. It's one of the most aggressive form of breast cancer. And I remember that day, I thought nobody should be sitting in that chair and told, there is a very high chance you're not cured. I was very lucky. I responded to treatment, and I went on one of the clinical trials. And even today, 2 years later, I still have 30% chance of recurrence, especially in these first 5 years. But I really hope I can see this little girl grow up. And I want that for every ALS patient. And I don't ever want anybody to sit there and hear there is no cure and we can help you just to live better. And with this, I want to thank the lab and every donor that gave us tissue and Packard Center that have made this possible. And this very amazing lady who came here 1 week before pandemic, the day I was actually diagnosed, and has been leading all this research. Thank you.
Thank you, Dr. Ajami, and thank you for the true human experience that inspires you. We appreciate it. Great. Wow, I'm inspired. I'm looking forward to our Q&A session. We have one more presenter who is joining us virtually today. He has actually diverted some of his travels. He's actually attending a significant ALS and Neurology Research Conference in San Diego, but he is joining us today via the wonders of technology that we've all grown so used to. Dr. Jeff Rothstein, MD, PhD, he's the director of the Robert Packard Center for ALS Research at Johns Hopkins University School of Medicine, and he's professor of neurology and neuroscience. Dr. Rothstein is a heavy hitter in the world of ALS. He has hundreds of publications. He has a very large lab. His work leads to many clinical trials that are tested, phase 1, phase 2, and phase 3. Our chapter is proud also to say that this last year we also leaned in, similar to Dr. Fowler, to support a large grant that we supported financially for one of his projects, which is an antisense oligonucleotide ASO targeting CHMP7, which is a little bit of some similar technology to what Nick Olney was talking about with tofersen. However, Dr. Rothstein's approach and his impact is so large that he's going to talk a little bit more about Answer ALS and the discovery of ALS disease initiation, repair of nuclear pores. So joining us virtually, we want to thank him for joining us remotely. And he's in San Diego right now, but Dr. Jeff Rothstein. So welcome.
Oh, thank you. Can you hear me? I want to make sure before I go on. Can you all hear me okay?
Yes, we can hear you okay.
Perfect. And sorry I couldn't be there in person. This is the largest meeting in all of clinical and basic neuroscience called the Society for Neuroscience, and hasn't been held like many meetings for several years, so it's sort of important, and it coincided with the flight to the West Coast. So at least I'm in your time zone, although I felt like I flew to Europe. It was such a long day. So this is going to be a bit of a change, and I apologize I missed some of the other speakers, because we're going to talk much more about basic science, but how that relates to ultimately clinical science for patients, because everything that we do for ALS has to start with basic science. And at some point we rely, of course, on pharma to help translate our work into eventually drugs and drug trials. And for that they typically come back to us, because we as a community know how to do that well. And I'm really going to talk about two projects, and so let me see if I can get into this. So I don't think this audience, I'm sure, doesn't need to know, so of course I always include a basic slide, but it's important to understand that ALS is not just the disease of motor neurons, the cells that control our muscles. It's also a disease of other cells in the nervous system. Although we try to study motor neurons, many other cells in the brain are affected. And in fact, we now know that up to 50% of ALS patients can have a very, very mild cognitive change, mild, nothing as severe as Alzheimer's or frontotemporal dementia, the second most common form of dementia. But there is a new gene, and I'll come back to that, in ALS that actually causes both ALS and dementia. But it is, as many in the audience know, a generalized disease, and to this day we have no effective cure. We have now 3 FDA-approved drugs, and hopefully some of that was discussed today, but I'm happy to discuss that at some later time point this afternoon. We do know that most of ALS is sporadic, happens out of the blue, and it's as much as we've done much more in understanding the inherited forms of ALS, as shown in this sort of timeline chart. And those the models that have derived from those genes, meaning when we find a new gene, we, by the way, is the royal we, not me alone, we generate mouse models or fish models and many other models, and those have led to discoveries of pathways, and that's shown on this side of the slide, many pathways which we've then tried, we or pharma have tried to drug, meaning develop drugs for, hopefully making a difference in ALS. However, one of the things we often don't know in patients, in fact we often don't know at all, is that although you can see in this graph, and by the way, I'm happy to share all these slides. There are many different pathways involved, but when we see a patient, we don't know, maybe it's oxidative stress, maybe it's aberrant RNA metabolism, maybe it's nuclear biology, which I'll talk more about, maybe it's all 3, or maybe one started a while ago, and by the time we see a patient, another is playing a role. And so in a given patient, we don't know we'll say the percentage of contribution of each of these pathways. And I say that because you could have a fantastic autoimmune drug or antioxidant, and it's the best drug possible, but maybe that injury is only 5% of the injury when we see a patient. And we don't know how to quantify that yet. Quite frankly, not just in ALS, but many other diseases. So the best we can do is try to understand each of those pathways and develop the most powerful drug. And in the future, and I'll talk about that with Answer ALS, try to find out when we see a patient, which is the right drug for the right patient, which is the dominant biological pathway. Or is there one that really starts it all off? One of the things we learned about years ago, and I apologize if I'm going to move fast because I want to cover a bit of territory, is that there's a protein that's normally in the nucleus, and it's called TDP43. Means TAR DNA binding protein, an old name, but it's normally in the nucleus of cells. But in ALS, in fact, almost all of sporadic ALS, it is no longer in the nucleus, but now becomes found in the cytoplasm around the nucleus, and it becomes aggregated. And that's shown in this cartoon. Normally it's very nuclear, where it has to do what it normally does. It regulates RNA and DNA metabolism. That's essential for cells, but it moves out of the nucleus or doesn't get incorporated into the nucleus properly. It's in the cytoplasm, it's aggregated, eventually cells die. And this is a hallmark. This pathology is found in essentially 98% of all sporadic ALS and most, but not all, familial ALS. Now, by the way, it doesn't mean that every cell has this problem. Only some cells have it, but it's a common footprint of an injury that we to this day didn't really understand. There's a lot of work focusing on that because it's very common. In fact, and I'll come back to this later, one of the earliest findings we know from rare studies where someone actually had a brain biopsy done, later developed ALS or dementia, and we know the first step that occurs is this nuclear clearing and then later the aggregation. So we have some sense of the sequence of events. And so I'm talking about basic cell biology. It's important to take a step back and understand what I'm referring to. Every cell, and that's shown here in this 3D cross-section, has a nucleus, and that nucleus controls all of cellular activity. It's where your DNA lives. The DNA gets converted to RNA, and that RNA eventually leaves the nucleus through nuclear pores, little holes. Think of this as a tennis ball with a bunch of little holes in it, and that nuclear pore complex allows things to move in and out of the nucleus. It is the master regulator. And it's actually the largest protein complex. It's not just one. It's made up of 1,000 proteins arranged in sort of a donut-like shape, some on the surface of the nuclear pore, the nucleus, some inside. So it's got a well-described structure. And it's actually known that mutations of some of those proteins cause a variety of different diseases. So a critical component of the cell. The reason that became important is because we began studying the biology of ALS, not using mouse models, because we've used mouse models as a community for many years. It was the first important tool. It's not been a tool, if we're honest, that's led to a lot of drugs, successful drugs. We've run, although we have 3 FDA-approved drugs, and we all know that's great, they don't cure the disease. It's an advance, but I can tell you there have been many, many other trials, in fact, based on some of those mouse models, that all failed in human. So doesn't have a high throughput in producing information we really need to help my patients and all of our patients. And about more than a decade ago, a new methodology evolved using blood cells or skin cells from patients, so actual humans, our patients, and we can convert those cells into what are called stem cells, very primordial cells that our bodies form from, and then we can re-engineer those cells into actual brain cells. In doing cells, and these are known as IPS cells, or induced pluripotent cells, they became potentially a true model of our patients. They're like the equivalent of a biopsy of patients, which, by the way, we can't do in the nervous system because your nervous system cannot regenerate itself, has no regenerative capability. Unlike when you cut your skin, if we do a biopsy, it heals. Your brain can't do that. But these cells became a foundation for truly studying ALS for about the last 8 years or so. And some people in the field have pushed away, I'm being a little exaggerating here, pushed away using mouse, but saying, you know what, this is a disease of humans, and I need to use a platform that truly studies the biology of humans. And we did that back in around 2013, 2011, when a new gene was discovered, and it was on that slide called C9orf72, or C9 for short. The C9 mutation is the most common inherited mutation in all of ALS. It's actually the most common mutation in all of frontotemporal dementia, the second most common form of dementia. And it's even found in patients with sporadic ALS, with no family history. And for that reason, many of us, maybe not all, routinely test all patients that show up in our clinics for the C9 mutation, in fact, now for all ALS mutations, but especially C9 because unexpectedly it can be found in patients with no family history, and we are as a community developing drugs to deal with C9. Well, the reason I point this out is because by having those real cells, those IPS cells, the motor neurons from patients that's shown here, we began to learn almost a decade ago that there was a problem in the proteins involved in nuclear transport, the nuclear pore. This cartoon shows that the nuclear pore is important for bringing proteins in and bringing RNA out. And one of those proteins seems to be aberrantly aggregated in this model system. Now, for what it's worth, being a neurologist and a basic scientist, wearing both hats, I know very well that models are interesting, but if I can't find evidence of what I see in an artificial model, be it a mouse or these IPS cells, in my actual patients, I'm going to be a little crude here, I consider that bullshit. Meaning I need to prove that what I see in a model really reflects what I truly see in patients. And to do that in these early studies, we found that these abnormalities found in this model system of human spinal neurons, the very cells that die in ALS, can be found in actual brain of autopsied patients. So there's clearly a relationship there. That gave us an early clue that there was a problem in this transport process, but we began to dig much deeper. And I told you before that the nuclear pore is made up, and I didn't tell you this, but every cell has a nucleus, and every cell has at least 2,000 nuclear pores. That's these dots shown in this slide here, if I can get my cursor to work, these little dots. And the green in each panel represents an antibody that we've stained to identify, in this case, TPR, which is a component of what's called the nuclear basket. NUP62, which is in the middle of the nuclear pore. It has a gel-like state in the middle to help control what comes in and out. Or those proteins that bind the nuclear pore to the actual, if you will, the surface of that tennis ball, the nuclear membrane. In this case, it's called POM121. And we began to look at the actual nuclei from these iPS cells, again, derived from our patients. We saw that there were about 7 of them were dramatically missing. So you can see POM121 in sporadic ALS is really missing. In fact, these 7 or 8 different nuclear pores were dramatically lost in both C9 ALS initially, but now we've seen them quite frequently lost in sporadic ALS as well. So C9 is a familial form, still relatively uncommon in the grand scheme of things, but sporadic, that's the heart of what we're trying to eventually understand, sporadic ALS. So we saw that they were missing, and there are many other studies I'm not getting into, but they were missing. And because we saw they were missing this model system, we went back to say, are they actually missing, though, in real brain, the nuclei from real brains of patients? And the answer is, you bet. And the arrows point to the fact in spinal cord neurons, the motor neurons, this POM, some of these nuclear pore proteins are dramatically missing, as well as in the motor cortex. These are 2 affected brain regions in ALS, whereas what's called the occipital cortex, that's at the back of your brain, and that's for understanding vision, and it's unaffected in ALS, and was unaffected in this biological defect, whatever that defect might be due to. So we were identifying a new biological defect. And by the way, I'm leaving out many other studies where we show that defect is not just an interesting picture. Functionally, these pores are actually not working properly. So what for cartoon, to summarize many of the things we learned, this is the nuclear pore, this is the part that binds it to the nuclear membrane, this is the inside, we've cut it in cross-section. We learned that one of the critical ones was called POM121. It binds the nuclear pore to the nuclear membrane, and when that one alone, just that one alone is missing, all those other 7 or 8 disappear. So it was the critical nuclear pore. And so when it's missing, it's reduced. The entire pore is abnormal. The transport, the function of the pore, moving things in and out of the nucleus was abnormal. And that ultimately led to injury to the cells or susceptibility to injury. And by the way, in C9 ALS, if we use a drug, and you heard this earlier, it sounds like about the antisense oligonucleotide that's been quite successful for SOD1 form of ALS, that same kind of antisense we began developing for the C9 form of ALS, and that antisense, at least one version of it, very quickly repaired the pore. So we knew this was a very early injury in C9 ALS, but equally, we felt it could be an early injury in sporadic ALS. Well, if the pore is defective, then the next question has to be, why? What's causing this injury to the nuclear pore? Why are we losing bits of the pore? And it turns out cells have a mechanism, and it's called escort3. And it's a mechanism that monitors various membranes within the cell, but a subset of that pathway known as CHIMP7, or CMHP, you heard it, we call it CHIMP7 for short, is involved specifically in monitoring the nucleus and the nuclear pores to find out if there are any problems with it, to grade them, to repair them. And so we began to focus on CHIMP7, and this cartoon just sort of summarized what CHIMP7 normally does. CHIMP7 normally lives in the cytoplasm of a cell, not on the nuclear membrane. With some kind of injury, CHIMP7 will move to the nuclear membrane and the nuclear pore itself, brings in a few other proteins of interest, and eventually the nuclear pore proteins, as you saw in ALS, are removed and undergo some degradative process. That's work that actually was discovered in yeast. Believe it or not, we use yeast and flies and all kinds of other organisms to understand the biology of cells. But no one really knew of anything of its biology in real cells of the human body, specifically the nervous system. So we began to look at that. And when I told you we make these iPS cells, when we grow them, we make them, and we actually start with a stem cell, we convert it into motor neuron, and we let it live for weeks to months. So we actually know the timing, just like we as human beings, we start as little babies, young toddlers, even older, and adults. And we can age these cells and study them at different ages for what it's worth, just like we actually do in mice. We have a newborn mouse, a young mouse, an old mouse, so all of us. And knowing that temporal relationship over time is important because ALS is a disease that doesn't start right away. It starts over time. And so when we began to look at these systems, we learned that at and I'm going to get a little specific here, day 18 is a time when these are motor neurons. There's absolutely no injury to the nuclear pore. And yet, even before there's no injury to the nuclear pore, this CHIM7 is now enriched inside the nucleus. Here's a control cell. It's not very much in the nucleus, but in C9 and sporadic ALS, it's really enriched well before there's injury to the nuclear pore. Nuclear pore injury occurs weeks later by a month of age, day 32. So it's an abnormality of just CHIM7. It doesn't tell what it's doing, but it's an abnormality of it. Again, if we look in human brain, do we see the same thing? You bet. If we look in cortical neurons or spinal neurons from our patients, those donated at autopsy, we see CHIM7 is apparently localized to isolated nuclei. And finally, we can actually look at histology from the brain, and you can see here that CHIM7 normally is mostly in the cytoplasm. These are large cortical neurons from autopsies. But here in C9 and sporadic ALS, that protein is now very enriched inside the nucleus, just like we saw in these iPS cells, thus matching the model system to the disease as well, the human disease. Oops, gotta get back on my slide. If that's the case, and again, I've left out some other studies, we know that CHIM7 is important in the degradation of nuclear pores. It is responsible for that loss of nuclear pores. For the sake of time, I've left some of that out. The point is, if that's the problem, if that's starting the injury to the nuclear pore, what if we get rid of that protein? And it sounds like you've already heard about antisense oligonucleotides. It's a gene therapy that very effectively and quite specifically can degrade targeted proteins. It's highly specific. It beats any other drug that we normally take by mouth. Really works quite specifically. And with the collaboration of a company that's very good at making these antisense called Ionis Pharmaceuticals, we could show that at least in this model system, we can really get rid of that protein. We can treat those spinal neurons with this antisense, and we degrade it beautifully. So we're removing that CHIM7. Now, for the sake of time, I'm not showing. We can deliver that same antisense to mice in the same way in vivo, meaning in a living animal, degrade that protein. And by the way, it has no toxic effects to get rid of it. That's important because if this eventually is going to be a drug one day, we want to be sure that we don't have any aberrant toxicity from getting rid of a protein. And those are very important studies when we move from the lab to the clinic. By the way, we found other methods to get rid of CHIM7 because you got to think in the future maybe antisense won't work as well. We have another gene therapy way of getting rid of it, and even another way using antibodies, very similar to some of the newer therapies in Alzheimer's to get rid of it. So different ways. So right at the beginning, even before we go to patients, we're finding different ways to hopefully get rid of the protein. Of course, the question is, does getting rid of the protein do anything? Does it fix the nuclear pore? Again, here's the nuclear pore. CHIM7 is aberrantly localized in the nucleus. And shown here are nuclei, spinal neurons, the same neurons in our patients, from controls. And you can see CHIM7 is not in the nucleus normally. Here are the nuclear pore proteins that should be on the nucleus, and they vary on how much we can detect. C9 ALS, now we see CHIM7 is localized to the nucleus, and those nuclear pores are really abnormal. Same with sporadic ALS. And by the way, although I'm only showing you an example of one, our experiments in the lab are never in what we call an N of 1. We don't look at one patient. Just like in the clinic, to prove something, you need many patients. And these studies were based on 30 different sporadic ALS patients. It's essentially just like an early clinical trial. So here's where we're starting. We then treat, once this injury is occurring, just like you see here, just like in a patient, we see patients already with an injury. We give those cells the antisense. Here we can show we lower CHIM7 very dramatically in C9. It's no longer really in the nucleus, also in sporadic ALS. And what happens to that nuclear pore when we treat with the CHIM7 ASO? We completely repair the nuclear pore. And in addition to repairing the nuclear pore, we completely repair everything downstream of that. Well, one of the most important things downstream, I started off telling you that TDP-43 is normally nuclear. it leaves the nucleus, or for whatever reason, it's not in the nucleus, and then it aggregates. When that occurs, there's a whole series of RNAs in the cell that are completely mishandled, and this is known as a volcano plot. But the important thing is, all of these dots above this line, they're abnormally low levels of RNA. These are abnormally high levels. And that means that the loss of this TDP-43 is causing a wide range of injuries to the cell. In fact, one of the first ones, the highest one that's changed, Stathmin-2, that alone is a future target that's going to start in this quarter as an antisense to try to fix that defect. Now, that's fantastic. That's exciting. We're starting to tackle these. But on the other hand, we're going to try to fix that one. Oh, but wait a minute. We've got all the other ones that we're not fixing with that ASO. Why is TDP-43 not in the nucleus? I've just told you for the last 15 or 20 minutes that maybe one of those processes has to do with there's a defective nuclear transport. So in fact, that's what we find out. The defect in the nuclear pore leads to a leakage, and that's shown here, of TDP-43, although some of it's in the nucleus, now some of it's no longer in the nucleus. That's a crude way of looking at things. It's what we call histology. But what about all those RNAs? What if we look at them, and we have, and this is a very busy slide, but what we're looking at is we're saying, when there's an abnormality in these slides, various of those RNA species are poorly handled. And that's the high bar here, the low bar there, the high bar here. But, sorry, I said that the wrong way. The low bar here, the high bar there, the low bar, meaning most of these are low in the setting of a defect related to TDP-43. In these very spinal neurons are patients. These are sporadic NC9 patients. But if we treat them with the chimp ASO, we fix, and this is a handful of the ones we're showing, we basically fix all of those RNA species. So we don't fix just one, we fix them all because we repair the nuclear pore in sporadic patients. And this is a real-world example. This is actually one of my single patients. So I've shown you a summary in that other slide. This is a single patient, don't know what's wrong with them, has ALS, and that patient has a defect in CHIM7, a defect in the nuclear pore, and a defect that's completely repaired by the antisense. We've now studied this, and I'll show you a slide in a minute, in more than 100 different IPS lines. Those IPS lines are individual human patients. They are our patients. It's like doing a clinical trial, and uniformly, the CHIM7 ASO repairs this defect. It's a model system. It doesn't mean it will work in humans, but it's the fundamental defect in all of sporadic ALS. And it's a fundamental defect we can actually see in what we'll call the biopsies from those patients. So what we've learned is that there's a defect early on in CHIM7, this screening protein, for whatever reason, is moving into the nucleus. It's damaging the nuclear pore, that's shown here. That's leading to nuclear transport abnormalities, and most importantly, that downstream loss of the normal function of TDP-43. And this is just a cartoon that shows that same biology. So we now have a new pathway, very common to sporadic ALS, an understanding of how that pathway is defective, and probably most importantly, potentially a drug that can repair that pathway. And to be honest, we're now working closely with Ionis to bring that antisense, just like you heard earlier with the antisense to SOD1, to patients as quickly as possible. There are a number of steps that we have to go through to prove it's still safe. It's still not a large animal. We're still testing it in animals and working out all the right conditions to bring it to patients, but I'm very anxious, hopefully, to bring this to patients. And that brings up the last part of the talk. All of us in ALS, those that try to understand the biology of ALS, have long understood that ALS is not one disease. Yes, it has the same name, but clinically, there are all different forms of ALS. There are patients where it starts in the speech muscles, what we call the bulbar muscles, the limb muscles. There are slow forms, fast forms. And so we lump everyone together for clinical trials, but that may be a mistake. And I showed you at the very beginning, there are all these different pathways in ALS. How do we try to understand that so we can truly personalize the approach to ALS instead of lumping everyone together? And to be fair, in medicine, this has been quite successful in cancers, in part because they can biopsy. So when I was younger, breast cancers are all treated the same. You gave a woman, you did a biopsy, you said, oh, look, that's cancer. And everyone got the same chemotherapy, and it really didn't do a very good job. by analyzing the biopsies and learning the molecular pathways, just like I showed you here in those previous slides, we began to learn that some women have Herceptin receptor positive breast cancer, got a specific drug for that kind of breast cancer, and did far better. And that same approach is being used in melanoma, various other tumors. And so that personalization based on understanding the actual mechanism relevant to the patient is critical. So how do we start doing that in ALS? And so this is a program I put together, started in 2013, but it began in earnest in patients in 2016. It's a program called Answer ALS. Now, you hear about Answer ALS, they're a foundation, but the name of this program is a Packard Center program. They actually give us some money to pay for this program because it's a really big program. And it was designed to enroll 1,000 patients at 8 sites around the country, and they're shown here on the slide. And they were distributed around the country. And then basically we chose these sites because we wanted to enroll patients very fast. The faster you can enroll, the less expensive it is as a trial, because this was completely funded, almost completely, by philanthropy. And some support from ALSA and MDA and a few other nonprofits, but not NIH, mostly philanthropy. And the idea was we'd follow these patients for 1 to 2 years. Not the idea, we did this. So we collect deep clinical data, not just, they have ALS or they don't, but different kinds of ALS. Every patient was given an app that we designed in-house by actually ex-Microsoft programmers, kind of out your way a little bit. So at home, because we know patients don't always make it to clinic, at home the app collected voice data and movement data and even a surrogate for breathing and cognition with the help of IBM Watson. Every patient's blood was sequenced for their entire genome through New York Genome Center, and all the blood was either banked at Mass General at a place called Neurobank. And to be fair, today we have more than 60,000 blood samples from our patients, a fantastic resource, and not just one time point, but multiple time points. That's important when you study disease. At Cedars-Sinai, that blood was turned into those iPS cells I told you about that we've built everything around. Those iPS cells are turned into motor neurons. We're talking about over 1,000 patients' biopsies. I call it a biopsy. And those cells then underwent a comprehensive biological analysis. Their DNA, all the RNA, all the protein, how the environment affects our DNA, that's called the epigenome. All of that data was stored in the cloud, in Cloud Azure, with the free help of Microsoft, who provided free storage space. And all of that data was designed, and the entire program I designed so that all of it would be freely accessible to both commercial and clinical and academic entities to use it to try to understand, to integrate that data through tools like AR, artificial intelligence, to understand, are there subgroups of patients? Because we now have that biopsy that was used in cancer. And we have all the deep clinical data to try to understand, are there subgroups of ALS patients that exist? And because if you think that there are, you've actually got the living cells to go back and test those ideas, and even the blood from patients to test if there are biological markers in the blood that relate to those pathways that you think, you of course, the royal we, the community. This is just an example. This part of the trial ended now about 2 years ago. We've fully enrolled patients, the fastest ever to enroll. They're the sites distributed around the country. Sorry, I know it's a little East Coast centric. That's us on the East Coast. What can I say? And here's the examples of what are called the functional, the ALSFRS functional curves of patients. You can see some patients drop very fast, unfortunately, and some are very slow. What's the biological underpinning to that? And can we target drugs that pick out this curve from another? In fact, our AI analytics team found there are at least 7 different curves of patients that flow during the course of the disease. As that means there are 7 different pathways, or there are different pathways for the beginning of the disease rather than the end of the disease. All of this data is useful for that. And the data is an example of the portal that recently we shut down, but we're going to open up again. We have to do a variety of things. But both academics, qualified academics, and qualified companies can go to the portal. They can filter for any clinical indice or biological indice, meaning various mutations or biological changes. And from that, they can select the data, download the raw data, or any or all of it for their own in-house research, or those iPS cells, or the blood specimens. So it's a huge resource to understand ALS. And I'll end by, actually before I end, this is just a little older summary. It's been shared by hundreds and hundreds of labs in the United States, Europe. You can see this map. And although all of the patients are enrolled, we haven't finished gathering all that data. The data takes longer to generate from the individual labs that are part of this big consortium. By the way, this is a consortium of more than 100 different investigators around the country that came together. So people sometimes think that we don't collaborate or in ALS. This was a cherry-picked, highly collaborative team that's worked together for this program. I say cherry-picked because, yes, if I'm honest, there are some people who don't collaborate, but you don't work with them. You work with only people who collaborate. And this is a fantastic collaborative team for that data. This is an example of what comes from that. So I showed you before, this is a very, on one hand, it's sort of a busy slide. Those are those TDP-43 abnormalities I showed you earlier. This is almost 100 individual sporadic and C9 patients compared to control. And this is all those TDP-43 abnormalities. The darker the red, the more abnormal. And this bar, all of them have a defect in nuclear transport. So you can easily see, just looking at this, it's almost black and white, it's red and pink, that sporadic ALS has a huge change in TDP-43 versus control, and it's all linked to nuclear pore and CHIMP7 defects. So this is an exciting direction for us. By the way, this is an animation of the nuclear pore itself. As I said, it's made of 1,000 different proteins in this wonderful puzzle. And we actually have clues that one of the defects, even in sporadic, may be there's some slight changes in that puzzle, allowing a slightly different conformation, activating this very pathway. So I think I just want to then summarize. We now know the nuclear pore complex is damaged in sporadic and C9 ALS. POM121, one of those nuclear pores, appears to initiate the defect. We know that CHIMP7 is critical for this pathway. It relocalizes the nucleus, and antisense is very much a treatment approach that we think could be important in the future for sporadic ALS. But there are certainly other ways. And by the way, TP43 is a defect also in FTD and Alzheimer's, so this could bleed into other diseases for future therapies as well. I didn't get a chance to really mention much of the recent work was done by then a very talented postdoc in my lab, Alyssa Coyne, who's just become faculty at Johns Hopkins, but of course a team in the lab, but none of the work I do. And I want to stress how important it is in those of us who do both clinical and basic ALS research, that this is an entirely collaborative group of basic scientists at Yale, Salk, Rockefeller, industry collaborators, the group at Ionis, and the group from Answer ALS, distributed mostly here on the West Coast with Clive Svensson and Leslie Thompson. And of course, no one funding organization made all of this happen. Multiple groups have come together to make sure we can move forward quickly in this program. And I'm going to stop now. Thank you.
Thank you, Jeff. Stay on the screen there, and I want to invite our other panelists to come up so that we can have a great dialogue and expand on some of the concepts that we've talked about today. And if the staff can circulate and gather questions, anything that's been written down and tables, and bring those forward, I'd appreciate that. So just raise those questions up, and we'll bring them forward. So I'm going to jump into, I think one of the things, Jeff, we really appreciate you sharing about CHIMP7 and ASOs, but very excited to see that overview of personalized medicine. And also amazing to see the collaboration that it takes. So Jeff, joining us on the stage is Dr. Nick Olney, who is the medical director at the Providence ALS Center here in Portland, and Dr. Bahareh Ajami, who is with the Oregon Health Sciences University, and Dan Fowler from Rapa Therapeutics, who talked about his T-cell therapy work. So, great. So I think one of the things that's interesting to me is we had people here today talking about understanding the etiology of the disease and the mechanism, and then talking about the design of potential interventions in the development of assays, and then bringing it forward in clinical trials, whether they're standalone or the Healy platform trial. I'm just curious if any of you could comment on the time frame it takes to go from sort of basic science into actual available therapeutics. And so I'll open it up to anyone on the panel.
So I can speak to SOD1. It's a great example. So it's well known in industry, and others on the panel can address this, how long it takes for a small molecule meaning an aspirin-like molecule. It's often at least 10 years, and there's various numbers at a cost of $1 to $1.5 billion. Antisense SOD1 is a great example because the SOD1 gene was discovered in '92. We built mouse models around that. By 2000, 2010, Tim Miller, Richard Smith, and Don Cleveland really began the first antisense studies in mouse models, purely to say, does it work? And that chugged along for, Tim knows it better than I do, but let's just say 6 or 7, 8 years. By 2011, 2013, the first antisense to SOD1 trial was run. That seems a long time ago. And the reason we didn't get to a final result earlier is because the company reformulated, made a better version of it. So we had to reboot that trial. So that first antisense trial was interesting, but it was what's called a phase 1. It wasn't really a clinical efficacy trial. And then we followed by the later trials, and probably less than 10 years for that antisense. And the reason I'm bringing this up is because there's a Stathman trial coming up, Stathman 2, which is one of those TDP targets. In 2019, the basic science paper of that target was published. It has to do with axon regrowth and biology, published by 2 major labs, a group in Harvard and a group at UCSD, and Harvard as well. This quarter, that clinical trial will start. 2019 to 2022. That's incredibly short from moving from fundamental basic science to actually a in-human trial with a brand new drug. Now, when a drug exists already, let's just say aspirin, of course it moves faster, but to be fair, I hope I'm not dissing anyone on the panel, most repurposed drugs don't really work that well. You're really trying to target a basic mechanism. That's not to say we don't want them, and they do move faster, but they're not really optimized for the right patient population. Sorry, I overspoke. Anyone else on the panel should, of course, answer the question, too.
Great. Dan?
Yeah, I can comment. Jeffrey, this is Dan Fowler from Rapid Therapeutics. Good to see you from a distance. Just to introduce myself a little bit, I spent 27 years over at the National Cancer Institute doing stem cell transplant. I know some of the guys over there at Hopkins. And then 5 years ago, retired from NIH to start a small T-cell therapy company in Rockville. One of our funders developed ALS. After I left, we were just going to do cancer, and that was our motivation to honor him and develop a program, a little bit of an offshoot from what Stan Appel has been preaching for 30, 40 years. I met Stan. We're taking a little different tact. We're doing inducible Treg cells. They're a hybrid of a Treg and a Th2 cell, so a little different functional phenotype. But getting to your point, Lance, about the rapid rate of clinical translation, I'm also a little more optimistic that as we develop capacity, it's easier to get things into the clinic. And cell therapy is actually maybe even more rapid because, for example, in our case, we started doing research in 2019 on patients' blood samples. We developed an IND, and now we're into our third cohort of a phase 1 trial, seeing some nice biomarkers and hoping to do a registration trial in the next year or two, depending on funding and so forth. So I'm pretty optimistic. And cell therapy is something we do our own manufacturing, so we're kind of in control of all the variables. And you can, each patient is treated with their own immune system. So each patient's immune system is their own drug. So it's a little easier to innovate and make changes to the manufacturing. I don't know if that answers some of your questions, but I would be a little more optimistic also going forward that advances can be made and iterated upon. A lot of it is a platform and capacities like you're talking about with your networks and so forth.
Great.
I guess I would just add briefly that I think I always try to add when someone's interested in clinical trials about telling them what they're eligible for and how much do we know about the specific target versus is this more a broader mechanism or, as Jeff hinted at, more a repurposed thing that might show some neuroprotective thing in other disease groups. But I agree that those ones can move pretty fast, but then they are a little bit more probably risky. And these great targeted drugs like the ASOs, those have taken a long time, but they're getting faster and faster to getting from kind of the bench to the bedside.
Great. Thank you so much. This is a very I think a good sort of question that sort of illustrates the experience of each person living with ALS and their own experience, but then when we hear terms like neuroinflammation. So if ALS is an inflammatory disease, why does my MRI present as normal? And it sort of asks that question about what you're looking at in science as inflammatory versus what's measured in sort of like clinical testing. So either probably Dr. Olney or Dr. Rothstein are probably good for that question.
I was about to say Rick. I'm sorry, I knew your father, but you go for it. I'm happy to add.
Yeah, so I mean, I think what we're talking about is probably a more, especially from our talks earlier, I mean, these are probably talking about really focal changes in the immune system. I think we have big studies of steroids that when you totally wipe out the immune system, that does not work for ALS. And so then, and I think also what some of this question is getting at is how we actually make the diagnosis of ALS. So this is a diagnosis of exclusion where the imaging will typically be negative, looking for any evidence of inflammatory lesions or any kind of other disconnection syndrome that we see on the structural MRIs.
Great.
Yeah, and I'll add, so I want to make very clear, ALS is not an autoimmune disease. We as neurologists know others. And it's well known that every neurodegenerative disease, ALS, Parkinson's, Alzheimer's, they all have an inflammatory response to the degenerating brain, whatever is degenerating. And that's well known and is a very popular target right now. The autoimmune approach was the most studied approach in ALS ever, all through the 90s and part of the 2000s. There are more immune modulatory drugs that ever in ALS than any other drug category. They may not have been the right ones, but they've been heavily studied. One of the critical factors often is that if the drug doesn't get into the brain, that's a problem. And so the drug's got to, depending on your theory, the drug's got to get to the brain to work. And that sometimes is a holdup for sometimes drug companies.
Great. This is a question for Dr. Ajami. Can you talk a little bit more about the study of, you're looking at ocular motor neurons, and has there been any sort of opportunity to look at this in people with ALS who are locked in with declining eye function, to your knowledge?
So right now, what we're doing, it's the autopsy that we have from patients from ocular motor neuron versus the lumbar. One of the problems in the field is that basically we don't have PET ligands to be able to look at the patients when they are alive. So we're heavily relying right now on the postmortem tissues. And going back to the previous question also about the MRIs is again, and one ligand is actually for microglia, which is not very specific. So at this point, all we're relying is on autopsies, and we just comparing ocular motor neuron from patient, like postmortem patients. That's what we're doing with the Packard Foundation. Thank you, Jeff, for funding us.
I do love cross-pollination.
Yes.
Yeah.
I do too.
So I know that Jeff and Nick, you both have met and know Dr. Richard Bedlack at Duke University. And he does an amazing work looking at alternative and off-label therapies. And most recently, he's done work looking at ALS reversals. Can you reflect on his study of ALS reversals and what that may mean to any individual person who maybe wants to see that as a path for themselves? Is it about the individual themselves, or is it about activities that they participate in, or what's your perspective on ALS reversals?
So Nick, I'm going to take this first because everyone's probably going to kick me out of the room after hearing my comments. So I've seen well over 10,000 ALS patients in my career. ALS reversal is not a real thing. I don't want to provide that for false hope for patients. In my career, I've had 2 patients who their disease have halted, and perhaps one who got better. 2 out of 10,000, one gives you a sense of, is that real or not? And more importantly, is it another disease that we just have not identified yet? And I know the history of ALS. Today, we look for a disease called multifocal motor neuropathy. It's one of the critical differentials. Many years ago, that was thought to be a variation of ALS. It's just that we learned more. And those rare patients that seem to get better, maybe it's possible they have a different disease. And lastly, historically, in 2005, there were patients with HIV who actually got better with antiretroviral therapy. And that was thought to be, oh, is this again an ALS-like reversal? But it was their HIV disease that caused an HIV-like phenotype. So I'm not here to take away hope for patients, but when you have 2 out of 10,000 patients, it would be I'm not going to use the word false hope, it's a bad term, but it would be just wrong for me not to educate patients about the reality of what the disease is.
Nick?
Yeah, and I would add, I think that I've not been able to look a deep dive into that data, but I do think that clinically, when I see a presentation of upper and lower motor neuron findings in one region without any other explanation, I'm pretty worried about ALS and preparing people for progression in the future. I guess for the specific reversals, I guess I'd kind of put it back to the trials. I would hope that if we get enough trials or cocktail of trials where we can significantly slow the disease down, then we could actually have some way of neuroplasticity where people could recover. But I still think we're working on that now. And I think for some of these reversals, I do question, was that initial diagnosis correct or not? And I think some of those might be those things that are the ALS mimics we know about and learn about in school.
Great, thank you guys so much. Back to a more basic science question. What, if any, use does CRISPR and gene editing technology have in the fight against ALS?
It's exciting. It's a really exciting approach. Sorry. I didn't mean to jump in. Literally, we have a group called NEALS. Many of the centers around the country are all part of this, and we evaluate as part of that organization companies that are interested in coming to ALS, try to educate them as best we can about a good trial design or what they need. And literally, yesterday, I was on a call, a bunch of us were on a call with a new CRISPR company who's got great plans. So it's an exciting idea for familial ALS. It's not yet sure that it's going to be relevant to sporadic, other than perhaps this like Stathman-like approach. It's unknown, but that's a neat technology. As long as we pay attention to that, we're not causing permanent harm to someone's genome. Those are going to be big issues. It's something that I think my residents and fellows can look forward to in the future is an exciting approach to ALS.
For us, it's a very exciting technology to be able to translate what we are finding in differences. We just, that's the next step for us. As we are finding genes between ocular motor neuron and lumbar, we're going back to see if we can actually CRISPR those genes in microglia and replace them. So in terms of the immunology, it's done really well, and we think it's just going to be very exciting for us to be able to manipulate the immune system with them.
It's interesting, CRISPR, antisense oligonucleotide therapy, there continues to be development of new technologies that can make a difference. What do you all see in the future? Is there new technologies that are going to enter the world of ALS research that can be similarly transformative? Sorry, I'm asking you to put on your fortune teller hat right there.
So one other thing that's important for people that may not know, it's the gene therapies are not just exciting now, but what's already happening is what medicine's all about. It's preventative medicine. So we, not me, but the community, we're already starting to follow patients who don't yet have ALS, but carry the gene, and maybe someone mentioned this before. I think it's called the ATLAS trial. So they're at risk, and we can measure things in their blood when we think things are starting to change before they have any clinical change. And as soon as that occurs, and by the way, I'm only talking about SOD1 patients, meaning those with SOD1 mutation, we're going to give them that therapy. So we're going to be essentially pre-treating a patient to head off the disease before it really hits them. That's phenomenal medicine. That's preventing, in hopes of preventing the disease from ever occurring. So there may be other technologies, and the panel certainly could describe that. To me, that's a real quick jump from, we don't even have SOD1 as a gene therapy approved yet, though I hope it will be, but we're already going to that next step of pre-treating patients. That's exciting.
Yeah, I guess I would echo that. I think that clinically, I think one of the things we're really trying to do is really promote the genetic testing, really, of any patient that's coming into clinic, just because these therapies are really coming online now. And then I think also focusing on pre-symptomatic is probably the next phase, so that we can roll in the effective therapies of the people before the disease is even manifesting.
Dr. Fowler, one question, part of this understanding who's susceptible to ALS is looking at potentially what a biomarker might be available. Is biomarkers part of your study, or do you see a role of looking at biomarkers? And can the rest of the panel talk about the importance of biomarkers in ALS?
Well, I would echo with what Jeff's talking about, is early intervention is going to be critical. I spent my career in cancer, and we all know the importance of, colonoscopies, mammograms, early diagnosis, and early intervention in a comprehensive manner. And that's where, ALS needs to go. With the research being done, you can envision a scenario where individuals are identified before some of the tragic loss of functionality, and I think that's going to be critical. In terms of biomarkers, yes, everybody's looking at different ways to quantify neuroinflammation or neurodegeneration with the neurofilament things. We're measuring these as well as a potential indicator. Are you reducing the degeneration? And if you can do that in the bloodstream, that's going to be very valuable in terms of how to monitor. Like with our cell therapy, we need to know how many doses of cells is any therapy is, especially chronic diseases. You don't usually reverse with just one dose of something. So we're going to need to address that, have biomarkers, have good readouts, and that's all things I think that'll be coming online.
Great. The rest of the panel?
I guess I would add to this practically. I guess just in being in a big healthcare system in the pandemic, I was just trying to figure out how to get patients even evaluated quicker. Something else we're working on. Yeah. I think my partner, Dr. Goslin, who's now retired, but she taught me that early on when I started my job here, of really trying to figure out ways to fight the grid. I'm booking out often 6 months in advance, but trying to prioritize my ALS patients, or certainly new patients, so I could get them correctly diagnosed and then potentially onto the right FDA-approved treatments, and then potentially if they're interested in trials.
Great. This is a question for Dr. Olney and Dr. Rothstein. Are there any studies looking at quality of life comparing the available treatments?
Off the top of my head, I would say I'm not aware of a specific study, but I know that some of these quality of life and actually looking at taking a lot of quality of life surveys of patients is becoming more popular within studies, but I can't remember a specific one off the top of my head.
Yeah, I agree as well. There's no question there's a great interest in making sure we pay attention to that and incorporating them in some of the studies, but there are specific studies, I think, being initiated. I can't cite them either, but I know through NEALS there's a number of groups interested in this.
For Dr. Fowler, is there a connection between your work with multiple myeloma, ALS, and diabetes? Would family history be an indicator of sort of connection between ALS and the opportunity to benefit from a potential therapy that you're working on?
There are genetic polymorphisms, so individuals who have a predisposition to get autoimmune diseases. That's been described in many settings, diabetes you mentioned. So I think in ALS, maybe the signal's not as prominent as some other diseases. So I view that as, well, maybe the drive toward inflammation is so strong. If you look at other diseases like Parkinson's disease, the amount of neurofilaments being released is a lot smaller because there's just a lot less tissue being. So I think we have to develop, targeted therapy is important, but you want to also develop treatments that are going to span across, and I think that's what Jeffrey, if you have a targeted therapy that hits very proximal in the cause of the disease, then that's a great thing. Similarly, if you have an inflammatory treatment that helps across all the different varieties, that's okay too.
Great. One question is, if there are many genes that are known to be part of ALS, is there an effort to look at genetic therapies for other genetic forms of ALS besides C9?
Oh yes, no question about it. SOD1 and C9 are the two current trials. Ataxin 2, which is a genetic modifier in ALS, is an ongoing trial right now. And then there's a new effort for really rare mutations, and the first one that's come to bear is FUS. FUS is a really uncommon mutation in ALS, and there's a trial organized through Ionis Pharmaceuticals involving many centers in the United States. And one of the interesting things about centers is a very patient-oriented trial, in that since it's so rare, it's not likely that a patient will live near a major medical center that's carrying out the trial. So the companies have arranged concierge services that bring the patient to the closest participating center, which is perfect. That's what you want. But the future iteration of that is something called NLORM, which is an effort for really rare versions of ALS, gene therapies, to be brought forward for these, what were called N of 1, meaning very, very rare mutations. And so there's a great interest in this effort tied to Ionis Pharmaceuticals for at least antisense for these rare versions. So yes, there's attention for those patients.
Great. This is more towards those of you who are treating clinicians. Could you talk a little bit about the new treatment that's been approved, AMX0035, and are you moving forward to work with your patients to get access to it?
I can take that because I talked a little bit about it earlier. I mean, I certainly am supporting access to it. I mean, I think the data looks solid. I reviewed just briefly some of those positive studies from the Centaur study. I do think it looks pretty safe. I think, again, as long as you talk openly about patients to educate them about the GI side effects, and I think I'm lucky to have a team of nurses that help me with all the advocacy to try to get patients that I do know. At least a few of my patients just last week have been able to actually get it. So,
great. Jeff?
Yeah, I completely agree. I'm excited to provide it to patients. Now, on the other hand, being a careful scientist, it's still only one trial. And if Nick didn't say this earlier, 80% of phase 2 trials, when replicated, do not replicate in phase 3, which means that follow-on trial may not replicate. If that's the case, stop the drug. But in the meantime, it gives patients access to the drug now. I see no harm in that whatsoever. There are other trials out there that are quite toxic drugs, not in the United States, but this is not one of them. So I'm excited by it. And I happen to know the two CEOs, what we call them the boys, these two young guys, and they're really good, and they care about making a difference. And I think probably both, Dr. Olney and I would both agree, it would be nice if it was a little cheaper.
Yeah. Yeah, I think that's good.
I would add, too, that as I mentioned earlier, my dad had been diagnosed with ALS, and I would actually be part of the ALSA National Advocacy Day at DC. We were always advocating for this early access to drugs. So I think this is kind of the day that has arrived that, you know, we think we're getting patients early access here, and I think you have to make sure people know that, too. But I think that's exciting. I mean, I've been advocating for years for that. So,
to be balanced, can you both also comment on the new oral version of Radicava that's now available and the impact it's having in your practice?
So Nick, I can start. So when Radicava first came out, it was the first, to put in perspective for all of you, we had gone for 20 years with only Riluzole, which is fine. People think Riluzole, all of these drugs, by the way, biologically, they're about the same efficacy. There's no huge difference. But it's good to have others. They work different ways. When the IV form came out, it was both really expensive and a very difficult approach to giving to patients. So there's an initial rush to patients, but in our clinic, it dribbled down to no more than about 5% of patients were getting IV. So to have an oral makes a huge difference. It's still a kooky schedule, if I'm honest, 14 and 10, but the daily schedule is a regular trial now going on as a phase 3 trial. So we'll learn two things. Can we give it daily, and will be equally effective or more effective or not effective? We'll learn all of that. So I'm fine with that. I think there was a bigger boost because of the Amylyx, but for what it's worth, there's probably more data on, not a lot more data on Radicava. And both drugs are relatively benign. IV form is not benign, mainly because you have to give it IV, but we're beyond that now. We can give it oral.
And I would add to that, too, just kind of echoing what I said in my talk. I mean, I think the earlier main side effects we were worried about, the IV Radicava, tend to come from the actual delivery and the infusion, and that was a real burden. Again, when I talk about it with patients openly, half or more would already be out the door before they'd even want to hear it once you mentioned IV. So I think the oral, I really like that it gives patients better access to it. I think, again, The battle now to get it covered is the other challenge, but those are things we're certainly working on. I think the dust is still kind of settling there.
Great, thank you both.
Yeah, notably, both drugs are very similarly priced and very ridiculously expensive. I'm sorry, I can't help but add that opinion.
Great. Thanks, Jeff. This is a sort of three questions came up, and I think this is they're linked together. So I want to sort of summarize them in a joint way. One is a basic question. If someone is interested in getting involved in a clinical trial, what steps can someone take to do this? Where do I sign up? The next question is, are there any trials available for people with 3+ years being diagnosed? And then the last question is, for people with ALS that are no longer eligible for clinical trials, what would you suggest for getting involved in research? So think about the continuum of the amount of time people have had the diagnosis.
I could take that one first. So, I mean, I think we're a big clinical trials group within the Northwest. There's also trials at OHSU, and there's up in Seattle. But I think talking to your, I always recommend, again, before I even talk trials, I say get plugged into your local team center. And then I talk about FDA-approved treatments. Then I talk about trials. And then I pretty openly tell people, I mean, that's why when you take the history from the patient, you try to figure out when did that first symptom really happen, because that really does determine, are they, well, one, kind of prognosis-wise, but also what trials or research they might be able to participate in. So as I talked about, the Healy trial allows up to 3 years. Most of them are typically around 2 years. I mean, even before that would be even more stringent. And I think for when it's over 3 years, I think that's when you're starting to think about expanded access programs, so certainly trying to look into that. And I know that one of our partner clinics, Swedish, was doing the expanded access Amylyx, and I would send Oregon patients up to there. I rarely sometimes hear some other trials that allow people more than that. I do think they tend to be more experimental as I went through the different phases, probably phase 1, phase 2 trials typically.
Great. Jeff?
Yeah, I agree completely with what Nick reviewed. There are websites also to consider. That is not, I mean, it's great if you go to a major medical center, but if you don't go to one, you may not have access to those physicians. And ALSA, MDA, and actually IMALS actually has a fantastic website for reviewing trials. Clinicaltrials.gov is a disaster if we're honest, because it doesn't filter them, and you can get a huge number. You need to go to a group that's paid some attention to the quality of the trials and the various domains, open label access, the things that Dr. Olney already went over.
Great. Well, we're coming to the end of our time today, but for those panelists who were here for the whole presentation, I would just like to ask all of you, what did you do you have a takeaway from your fellow presenters, and what did you find interesting, and something that gives you, sparks your curiosity or hope?
I'm going to start. As a basic science researcher, it's really interesting to see that it actually translates to drug, and especially hearing about Dan's talk and how fast it could now be translated. I'm very excited because we don't see that end of it. We see just the targets that we discover and mouse work, and then hopefully it's going to translate to something that would be a drug for a patient. So that was very exciting for me.
Great. Dan?
Yeah, I'm humbled to be here. Thank you, Lance, for your support. I mean, hearing your research on the microglial cells, I told you I have a slide or two that has a microglial cell line in there, so I was happy to see your, I didn't overstep my bounds. But it shows how researchers, you have to respect a 20-year career, whatever, where you've been doing deep dive, and I'm benefiting from that because I read the articles and I have access to that, and we try to compare and contrast. So that's very motivating. And then to hear Nick's efforts here and his training and your father's struggle with ALS and the loss there, it's very poignant to hear that.
Yeah.
And then to hear Jeff's maverick work with tying everything together and going after the root cause of ALS, and it's just amazing to see the progress. And then for me as a cancer doctor to try to be a part of that from an immune angle, coming in from the outside. We've been embraced by all the community and getting support. To me, it's very, very gratifying and challenging.
Yeah.
but thank you.
And Nick?
Yeah, and I would add, thanks again for inviting me to talk. And I think always seeing really cool basic science presentations is awesome, because I mean, that's the stuff where I think everything starts. And then I want to see that safety data. And then when you're done that phase 1 trial and you're looking to get 100 to 200 patients in, give me a call and we'll see what we can do. So, but I always put the safety data, really, that's really what you want to see. You don't want to obviously cause harm to patients, and going from there.
Great. And Jeff, final word for you. What gives you hope in today's ALS world for research and treatment?
So there's such a fantastic growth in both companies and academics, but especially companies diving into our field and trying to bring forth their creative creations. And there's wonderful working relationships now between corporate organizations, companies and clinical trialists. It's incredibly exciting. We didn't even get deep into the biomarkers. There are new emerging biomarkers that are being incorporated into trials. And as a group, we're working well together. And lastly, and quite importantly for the patient community, there have been so many changes in trial design to make sure we bring as many patients in as possible, to afford patients that opportunity to experience new clinical research and new drugs. I've been doing this for decades, so I'm extremely excited by what's going on now.
Great, thank you so much. This afternoon has been an amazing, illuminating, and edifying seminar. I'm always inspired when I hear the basic science research, like Dr. Ajami and Dr. Fowler, and how that moves into treatment and study and becomes therapies that can move towards becoming available. And I thank all of you who joined us online and all of you in this room who came together to learn and think hard. I thought hard as they're presenting their slides. And I'm also grateful that there are people who are a lot better had much better math education than I did when I was a kid. But we would not be here today for this seminar if it weren't for our sponsors, so I want to thank them again. So let me read them out again, and we'll give them all a round of applause, as well as our presenters. So in particular, thank you to the Providence Brain and Spine Institute, New Motion, United Access and Ability Center, Norco, Mitsubishi Tanabe Pharma America, Pharma, and Amylyx. So thank you to all of our sponsors. We deeply appreciate your support. And thank you to all of our panelists and Jeff for joining us after rushing off an airplane and getting into his hotel room. It looks like a nice room, Jeff. And our panelists are here, and I know they'd be happy to touch base, but also please take a moment to go around and visit all of our sponsor booths. Many people traveled here to connect, and we are just appreciative, and I'm appreciative of all of you for coming together today. Research is a huge part of our mission collectively as our community, and it's all of us working together that will find new treatments and new therapies. So thanks, everyone, and thanks to our panelists again.